Namaskar, my dear students. As you know, we started with the chapter Sexual Reproduction in the Flowering Plants last time, and in that very chapter I told you about the way in which the plants reproduce by the sexual reproduction, and I told you that flower is the main part of the sexual reproduction, is the main organ of the sexual reproduction. We discussed here in various parts of the flower and then the sequence of the events which take place during the process of sexual reproduction. This sexual reproduction is a continuous process, but for our convenience of understanding, we have divided the life cycle of the flowering plants into 3 steps, that is pre-fertilization events, fertilization and post-fertilization events. However, they proceed one after other in a continuous manner, and then we started with the pre-fertilization events, in which I told you that pre-fertilization events are those events wherein the flower undergoes some changes and prepares itself to cause the fusion of the egg with the sperm. And we have classified the pre-fertilization events into 3 steps: the microsporogenesis, which is the development of the male gametophyte, then is the megasporogenesis, development of the female gametophyte, and pollination, that is the transfer of the pollen from anther to the stigma. In the last topic, we discussed in detail about the microsporogenesis, and today we shall be starting with the megasporogenesis. The megasporogenesis occurs in the female reproductive part of the plant, and this female reproductive part of the plant is the gynoecium or the pistil, which is composed of one or more carpel. A carpel, as you can see in the figure, has 3 distinct parts: ovary, style and stigma, and in a highly modified leaf, and it is a highly modified leaf folded along the midrib. The margins of the leaf fuses to form the ventral suture of the carpel, and the midrib side represents the dorsal suture. This is, I am telling you how does the pistil develops from the leaf-like portion of the plant. The cushion midrib, the cushion-like parenchymatous outgrowth develops on the ventral suture, and that is called the placenta, on which the ovules are born. Ovary is the lowermost, this you can see, ovary is the lowermost swollen portion of the pistil, containing one or more chambers, which are called as locules, or in plural you can call them loculi, and accordingly, if it is one, the ovary is called as unilocular; if 2, then bilocular, trilocular, tetralocular, and so on, and if many, then it is multilocular. On the basis of number of carpels, the gynoecium, it is called as the monocarpillary if there is only one carpel, and polycarpillary if there are many carpels, and correspondingly if 2, then it is called as bicarpillary, tricarpillary, etc. Now, after studying the structure of the pistil, I will tell you that the main structure where the megasporogenesis takes place is actually the ovule, which is present inside the ovary. The placenta, as I told you just now, the placenta you can see in this figure. The placenta is a ridge of the tissue, a parenchymatous mass in the inner wall of the ovary to which the ovules are attached. The manner in which the placenta are distributed in the cavity of the ovary is called as placentation. That is the placentation, the arrangement of the ovules within the ovary. Each ovule is attached to the placenta by a slender stalk called as the funiculus. You can see here is represented the funiculus. Now, this funicle, that is And then the point of attachment of the body of the ovule to the stalk or the funicle is called as the hilum. This one you can see, this is called as the hilum. The inverted ovule, the part of the funicle, remain attached beyond the hilum alongside of the body of the ovules, forming a sort of ridge, and that ridge is called as the raphe. You can see here it is forming a ridge to which we call as the raphe. Now the ovule contains a mass of the thin-walled parenchymatous cell which is called as the nucellus. The nucellus is protected by one or two more multicellular coats, and these are called as the integuments of the ovule. The basal portion of the nucellus from where the integuments appear is called as the caleza. And then if there is one integument, it is called as unitegmic, or there can be two integuments, then it is called as the bitegmic. A small opening is left at the apex here. A small opening is left at the apex which is called as the micropyle. Female gametophyte or the embryo sac is embedded in the micropylar region of the nucellus. Now, after studying the structure of the ovule, I will tell you that this is not the only structure of the ovule, but the most common type of the ovule which is found in most of the families of the plant, and such a ovule is called as the anatropous ovule. Apart from this, we have many other variations in the structure of the ovules which you can see in this figure. The first one is representing the orthotropous, wherein the funicle, caleza and the micropyle are in the straight line. Second one is the anatropous, which is the most common and the one which you have studied just now. Then is the hemitropous. Third one is the hemitropous, in which the micropylar end and the funicle are at right angle to each other. Then the fourth one is the campylotropous, wherein the ovule is arranged in such a way that it is little curved. After the being at the right angle, it is little curved. And then is the amphitropous, and the last one is the cercinotropous, wherein the micropylar end, funicle and caleza again come to lie in a straight line, but after getting it round, after getting a twist of about 360 degrees, that is, it is turned inside out. And then these are the various types of the ovules, but the most common type, as I told you, is this one, which is called as the anatropous ovule. Now, how does the various changes take place inside the ovule? That is what I should say, that is the development of the female gametophyte inside the ovule. Herein you can see a series of steps in the figure. How does it take place? In the nucellus, towards the micropylar end, hypodermal distinct archaesporal cell is formed. This divides by periclinal division to form an outer parietal cell and inner primary sporogenous cell. Primary parietal cell divides further to form parietal tissue of the nucellus. The primary sporogenous cells forms megaspore mother cell, MMC. The megaspore mother cell undergoes meiosis to form the megaspore. As you can see in the figure, that the first from one cell the meiosis occur and the 4 cells are formed, and the megaspore mother cell undergoes meiosis to form the 4 haploid megaspore. This step is called as the Megasporogenesis. Out of the 4 megaspores in a linear tetrad, usually the upper 3 degenerate and the lowermost enlarges to become functional megaspore. That is, once the, as you can see here, that is 4 cells were formed, the 3 degenerate and 1 develops into the megaspore. Now the functional megaspore. Herein starts the development of the female gametophyte. The functional megaspore forms female gametophyte, or we can call it embryo sac. The nucleus of the megaspore divides into 2, 4 and finally 8 daughter nuclei, 4 of which are located at each pole. One nucleus from each pole, you can see in this figure, one nucleus, there are the 4 nuclei at each pole, and one nucleus from each pole migrates to the center to form the polar nuclei, which further fuses to form the diploid fusion or the secondary nucleus. Thus, the central cell bears the 2 polar nuclei. 3 nuclei at the base of the embryo sac form the antipodal cells. The remaining 3 nuclei at the micropolar end get surrounded by the cytoplasm to form the pyriform cells. These 3 cells together form the egg apparatus, which consists of 2 cells known as the synergids are the help cells, and the egg cell are the oospore. Egg cell are the oospore. Now, which hangs between the egg cell hangs between the 2 synergids, and the synergids bear a special type of the thickening at the micropylar end called as the filiform apparatus. Now, how does this figure appears? You can see this. Now, actually embryo sac, when it is fully developed, it appears like this. The upper side, the micropylar side, it consists of the synergids, one egg cell, and then the you can see the filiform apparatus inside the synergids, which play very important role in guiding the pollen tubes into the synergids. The egg cells on fertilization gives rise to zygote, while the synergids gets disorganized soon after the fertilization phase is over. On the other end, you can see 3 antipodals, which sooner or later also get disorganized. This may, however, be nutritive in function. Thus, antipodals may play a nutritive role, but ultimately they get disorganized. Thus, they may, however, be nutritive in function. Thus, the typical angiospermic embryo sac at maturity is 8 nucleated, 7 celled stage. Now, you have studied here in detail the process of microsporogenesis and megasporogenesis, or in other words, you can say development of male gametophyte and the female gametophyte. What were the various differences? You can enumerate between the two. That is, the male gametophyte is derived from the pollen grains or the microspores, which are formed in the pollen sac of the anther. Whereas in megasporogenesis, the female gametophyte is derived from the megaspore, which is formed in the nucleus of the ovule. In microsporogenesis, microspores are shed when mature and transferred to the stigma by pollination, whereas megaspores are not released from the ovule. In microsporogenesis, vegetative tube, nucleus and the 2 male gametes are formed, that is 3-celled, whereas in female gametophyte 8 cells are present, that is 2 synergids, 1 egg, 2 polar nuclei and 3 antipodals. Then we can talk about their function also. In microsporogenesis, one of the male gamete fuses with the egg to form the zygote, second fuses with the secondary nucleus to form the primary endosperm nucleus, which we shall be studying after this chapter, this lecture, that how does the fertilization takes place. Whereas in the egg cell, it fuses with one of the male gametes to form the zygote. One synergid degenerates and the second directs the pollen entry. 2 polar nuclei fuse to form the secondary nucleus, whereas whereas the 2 polar nuclei which further fuses with the second male gamete to form the triploid endosperm. The antipodals degenerate before or after fertilization. Now, This is all about the Megasporogenesis. The next time when we meet, when we shall be here with another lecture, we shall be talking about the pollination and the fertilization phase. Till then, thank you, stay safe, and my dear students, I would like to tell you that in case you have any query, you can leave your question at my email, or you can talk to me on my phone number given at the end. And moreover, because we are studying this chapter in a continuous manner, it is very necessary that you go through the previous lecture before you understand the next one. So I would suggest you to go through your books and the notes and the lecture so that you understand it fully. That's all for today. Thank you very much. Stay safe.